To talk to you is that history does matter, at least a little bit, and I've had a lot of experiences along the way, and the more that I could share those things with you and the more useful they are to you, I think the more everybody's better off. So do ask questions. But as I was saying just a minute ago, maturation is tough, you still think you're 20 years old and then you're nearly 70. That happens to people. But then I was like, I used to give the talks and I'd go, oh gosh, some of these people, they weren't even born when the Genome Project started. But now I'm thinking you weren't born when it finished, right? And it took 13 years. So the punctuation points, but 2000 was the draft and 2003 was the essentially finished genome. So was anybody born like after 2003? Okay, that's good, But maybe some of you were born after 2000 or just before, right? Yeah, so, well, then this is going to shock you, this, right? I'm going to show you one slide here. Let's see if that works. I'll have to grab the computer. There we go. So, you got no idea how horrible life was back in the 1980s, right? So, like, there was no computers, essentially. But you've seen some of these things, like, what Space Invaders are, right? It's the very first video game. Like, prototypes of the basic software that could make a thing go, like, it was really bad. And then, of course, no cell phones and no interconnectivity. That was like what was really hard to imagine. And in the lab, we had radioactive sequencing. You all know about Sanger sequencing, right? Well, the non-radio labeled methods haven't really been finessed. In fact, the Maxim Gilbert methods, which used like really super toxic chemicals, were still popular, right? So, nobody really knew how long molecular biologists lived or DNA sequencing people lived. The chemists would all die young, but the people were like, hopefully the molecular biologists won't have that happen. So, so it's pretty bad. And this is in the 1980s. And at that time, let's see, that like very few genes were sort of known and understood, about the hemoglobinopathies, right? And, a few genes that were really high on this disease list. And when people talked about the knowledge of genes, they didn't know them like you do. Like, about the sequence and what's known about the, things that activate them and how many exons they've got in the splicing. Some of it was conceptual. Like the cystic fibrosis gene was just a linkage spot, that ran in families. So it's all very vague, and you should be very glad that you didn't live through that period. So the Genome Project got underway by a few enlightened individuals who thought that, out of the box, thought, even though it's impossible now, wouldn't it be great if we had all that DNA sequence that through the whole genome? Actually, some of the major thinkers were influenced by the early radiation studies. The Department of Energy had studied radiation effects after the atomic bomb, just 100 years ago, and we're looking to see what the ongoing mutational burdens were, and wanted to build all the information around that. So they were thinking, like physicists, a lot of them, they thought big, but why don't we just sequence this whole damn thing and we can do it that way? So it took some time through the 80s for that to get traction and for groups to get together and to say, let's do it. And I guess hello to everybody online. I'm not sure who the audience is, but there are some certain various characters. And James Watson, who's completely been cancelled now, back then was very instrumental in getting money out of the non-NIH funds to work with the Department of Energy to get this whole project going. So there was a sort of goal set then, well, why not sequence the human genome? And various labs who had people in it that were sort of active enough in thinking that way, glommed onto it. There's different kinds of people. Some people wanted to find more disease genes. Some people were kind of digital people, although there weren't that many of them back then. And some were sort of techno geeks in molecular biology, and some were like yeast geneticists, very diverse group of people who all came and aggregated because money had become available to study that genome. So I'll stop there for any questions anybody wants to ask already, or I can just keep going. Okay. So myself, I was a born in Australia and went to Melbourne University and studied genetics and radiation biology. And I was interested in the relationship between lethality and cellular transformation, given different qualities of radiation. Actually worked with iodine-125 decay as a mutagen. Of course, a lot of double-strand breaks. Some of you probably dabbled in this area too, right? So DNA repair, radiation effects, cellular transformation. And so when I came to Houston, I was fortunate to come and work with a guy called Dr. Tom Caskey, who passed away 3 years ago. And he started the Department of Molecular and Human Genetics that some of you are associated with, right? And he did it with the idea that as a clinical, as a pediatrician with an interest in genetics, he could start finding disease genes in children in the TCH system. So he built that department around that, building basic science connected to that. And Tom was interested in the idea of genomics and attended a few of those early meetings and encouraged everybody to develop technologies, think about DNA, find variation, and use that to drive biology. So I came as a postdoctoral fellow in that period. And then just through my own meanderings and my interest in my own things that I was doing, I got I guess, engaged in the very early parts of the Human Genome Project and in DNA sequencing technology. Let's see if we can move this on because we're out of the 80s. Like I said, I can talk for hours, so don't hesitate to interrupt me. Or I guess the pizza will come, right? And that'll be the natural moment to stop. So, so when I came to Tom's lab, of course, we were interested in a disease called Lesch-Nyhan syndrome, which is HPRT deficiency. Have you ever seen a child with that?
Not clinically, but I've heard like other clinicians talk about that.
Yeah, some of these like ultra rare diseases, there's like 10 people in the world with them. That's one of them, but it's a devastating disease. But it's caused by HPRT deficiency, and HPRT, hypoxanthine phosphorybosyltransferase, is a cell culture marker you may have used. And you can drive it forward and backwards depending on which chemicals you put in the cell culture, and plus it causes a human disease. But nobody knew really the mechanism of gene inactivation. So like this is up there with no PCR and no telephones, right? We didn't know what broke genes. And so we developed methods to interrogate the gene. And a guy called Kerry Mullis came along and talked about polymerase chain reaction. And we immediately got the bug and said we can use PCR to amplify the gene if we have sequence. And then we can use the new sequencing methods to actually look at the DNA sequence level in those genes for patients. So this is all very routine right now, right? That's what you do. But back then we couldn't do it because we didn't have the sequence flanking the exons to make primers to do the PCR. And one of the things Tom did in that early days was formed a collaborative relationship with Wilhelm Anselby at the Embl labs in Heidelberg in Germany. And Wilhelm had developed one of the very first fluorescent sequencers that was called an automated sequencer. But in fact, it was so manual that you had to look at the screen and write down the color of the thing going past. So it was pretty primitive. But with some students here and people there, and Dr. Andrew Civitello, who's now the head of transplant over at St. Luke's, sequenced 44 kilobases containing the gene, which was a big deal, gave us the flanking sequence, and we're able to amplify all the exons and then use the very first fluorescent DNA sequencer to look at heterozygosity. So this is this sort of period here, advancing mutation detection, getting better DNA methods to go. Set the pizza. Tell me when you tell me, do you want to get the pizza right now? Take it. I'll give you 1 more minute because it'll get you out of here. So that really got us into the Human Genome Project because we figured if we could do this for one gene, why couldn't we do it for all genes once we had the whole genome sequence? And we could use the methods that were developed in that period. So we joined this endeavor in the early 90s to sequence a human genome and to campaign for the money and all of that stuff. So I'll leave that hanging and you can go and get your pizza. That's probably more on your minds in the next phase of the Human Genome Project. Okay? Better run for it or you won't get any. I think that's my advice.
Because I did undergrad last year, like a big discussion was so one of the major centers.
They're sequencing the word. What years were you, what year?
05 through 09. So,
Oh.
I mean, certainly the Human Genome Project has already came out, but then I think at the time, they actually had one of the most interesting classes I took when I was in college is that we're actually sequencing the dot chromosome on Drosophila, because I think they're really comparing a lot of cancer and I think the Hawaiian speech, trying to understand the difference between that. I'll say that's actually the most interesting concept of the non-graph.
Yeah, oh, well, Watson was certainly a major contributor to the genome, and Waterston was there until about 2005, before he went to Seattle. Yeah, so we had some good friends, Rick and Elaine. I don't know.
Yeah, I remember. Yeah, she was still there, like when I thought that was she, yes.
Yeah, it's interesting where they are now. So, these guys probably won't be very interested in this, but it's interesting to think about how we did so very well in clinical translation of genetics here, and they really didn't.
Yeah.
It took many years to get this. And the reason is that the pathology department there was very strong.
Oh yeah, I think that was one interesting thing we were talking about, because there's some rumor about how TCH wants to start their own sequencing stuff.
Yeah. Jim Austin wanted to do that
I
forever.
know, but then we were talking about it's like, why? Because we love Baylor Genetics, like really cool. And a lot of them are trained with us, and then we have naturally a way in with them. Like if we need specific things, it's easy to talk with the directors and all that stuff, and then we know their interpretation because they're trained to interpret those things. So
Yeah. Well, so there's a whole story about Baylor genetics that I could share, but these guys won't be that interested in. I'm going to pass on it. Thank you.
Used to be the legacy of
I'm doing Healthy January. You heard of that?
no, I've never heard of that.
Hear any weird sound? They said that something there really was a sound
But then the
very.
No,
Okay.
It was okay.
Okay.
Is it online working?
Okay, actually,
Is this about right?
Yeah, I think that's nice.
So like, actually, like 10 years ago, when it was still
I think
terrifying.
still sort of left the rallies.
Like,
The behavior.
What kind of feature you want? I can like grab that for you.
Yeah.
You mind if we have 15 minutes ish?
No.
They do, but like.
Because like that Italian piece of they insist on doing anything. They don't.
You've got to get it while it's hot.
Yeah. They insist on that. Yeah. So we have a 15-minute break, like please, but then we'll continue the whole day. Cool. Thank you.
Right now, we do have both.
Yeah, this. Yes, we have a microphone.
And like,
Yeah, I suspect there's an audience out there.
Right.
Yeah, well, there's a little, because there's lots of nuances in every story. Basically, we started a collaboration with Thermo Fisher Life Sciences, and they just died. And they had this funky thing that turned into the solid instrument. Remember?
Oh, yeah, I've heard that. That wasn't like none of that.
Are ready to go? Is that on mute?
Yeah, I think, yeah.
Yeah, he muted the thing, so.
Yeah, I mean, I'm already unmuted.
Okay, we're good to go. Okay, we're back into the history story. Don't worry, we're going to move through the decades pretty quickly now. So where were we? Well, we're just starting the Human Genome Project, and there was probably 20 groups starting to do that in different ways. And then that number of groups sort of collapsed down as the grants got more stringent and things happened at the national funding level. There's books about it, so I won't elaborate on it. And we moved through the period of the Human Genome Project. It wasn't without drama, because how many of you have heard of this? The private-public battle for the human genome. Yeah? Whose side were you on?
I don't understand.
Okay. So basically, you know, it was a publicly funded group, and our mentality was that we wanted to have this data be freely available. That is, we wanted people to be able to get the sequence really fast, which is actually pretty unusual for scientists, right? How many scientists do you know that will give you all their preliminary data without feeling anxious about it, right? But to a large extent, I don't think we all realized how valuable, not how valuable, but how accessible how useful the data would ultimately be in near term. In other words, it was so much data compared to what we were used to. We thought, yeah, we might as well put it out there because who can make sense of it, right? And but the kind of stated principle was we didn't want people to patent sequence. We called it a land grab, you know, where you go out and you stake out places to drill for oil, even though you're not going to drill for oil for 10 years. So we didn't want people to do preemptive patenting. And what happened was, you know, this is us all there's Watson on up to the back right there. And then we went to Bermuda and discussed those things and developed what are called now the Bermuda Principles of free data release. But about that time, a guy called James Webber with Gene Myers came to the meeting and said, you guys are crazy because you are cutting the genome up into clones, into back clones, bacterial artificial chromosomes, and mapping them. Why don't you just do a whole genome shotgun? And there was a lot of pressure back on that because we didn't have the technology really to drive that kind of deep sequencing. We knew there were a lot of repeats. We knew it was already hard to assemble these small clones. There was a lot of resistance, and we knew we'd get precision from our method if we went back by back. But a guy called Mike Hunkapiller, who was the founder of Applied Biosystems, which is like the first DNA sequence manufacturer that was really successful, looked at the work of James Webber and Gene Myers and modeling the whole genome shotgun and said, well, we've just invented this new sequencing machine that you no longer have to pour gels, like, you know, between the glass plates with the acrylamide. It has capillaries and is highly automatable and can generate sufficient data to do the whole human genome by whole genome shotgun. And we're going to start a company because their corporate situation allowed them to do that. And they got Craig Ventnor, who had another whole set of history of doing radical things that you know, shook up the academic community, and got Craig to start this company called Celera, whose mission was to sequence the human genome and then maybe have people access the data but pay for access. So depends on whose history you write. There was us sort of on the public side saying it's going to be free open access data, and a private group trying to do it with the latest technology. And so we actually had a fierce competition for a couple of years. And the result of that was that we came together at the end with some pressure from political pressure and said, well, we're kind of doing this all together, published 2 papers. One was the Celera whole genome shotgun genome. Ours was a back base but unfinished. In other words, we did the random phase of sequencing for each back and then put it all together and then called it a draft of the human sequence. So that was a 2000 product that we had. So do you have any questions about that? It's a bit of a good example of history and something you could read more about. Exciting times. People in, yeah.
Was it like, I mean, I heard a rumor about how it was kind of the pressure from Celera that actually then pushed like the public like back like the one that doing like the backloading to speed up and actually publish the genome. Is that true?
Oh yeah, totally. So you know the Cold Spring Harbor meetings, right? So we went to the May meeting in '98, and Craig came along, and flew in on a helicopter or a limo or something, and we're all summoned to the one of the rooms up there, a bit like this, and sat around and listened to Craig tell us that he had all this in the bag, and we should just go do the mouse. Right? So, thank you. And then he left. Actually, on the way out, he grabbed Jerry Rubin, who was a Drosophila guy, by the collar and said, "Come out here," and did a deal to sequence Drosophila. That's the first model for shotgun. But so we're all there saying, what will we do? Now, the Wellcome Trust, who fund the English aspect of this, a few phone calls, they doubled their money to put in. They said, we'll do at least half the genome if we'll pay for that. So they raised another, I don't know, 200 million pounds straight away to do that. And the NIH group had a similar sort of visceral reaction, but it took months and months to get grants and get programs into the right place. But indeed, the NIH group accelerated as well. And so we all met there and came up with plans and thought about how we could produce a product that would be faster. The debate kind of went on for a while. And the reason we had a draft in 2000 was because of the pressure from Celera. Also, the reason there were 3 NIH centers and not like 7 or 5 that were around before that was for the same thing. Let's concentrate the resources in the places that can really get the job done. So those 3 places were the Whitehead Institute, which is now the Broad, and in St. Louis at Wash U, and then here. And so we had a meeting here and we formed a group called the G5, which was 3 NIH centres plus the DOE representative and the guy from the Wellcome Trust in England at the Sanger Centre. And we talked about strategy and how we do that and got it done more quickly. Yeah, so you're exactly right. And that's the next part of the story. So, that White House meeting in 2000, we all went to the White House and listened to Craig and Francis Collins and Bill Clinton talk at the podium and announce this new map from mankind, and had cups of tea, and then we came home and spent 3 more years finishing the essentially complete genome. So let's go back. Well, no, I'm going to go back 1 slide just to for a moment, just to give you the rest of the history. So that was the Genome Project, but like I said, it took till 2003, which is just over 20 years ago, to get that essentially complete project. And I'm sure you all followed, like 2.5 years ago, the genome's finally finished, right? So the genome has been done several times. They're like the draft, 2003 product, the 2000 and what is it, 2022, right? Yeah. So depends who you are and what you did, which one do you think was the most important. But the 2003 product was pretty damn good. After that, we did the HapMap project. You're familiar with that? It was like the first genetic mapping project. The genotyping methods were pretty primitive. And so there was a program that assembled donors from different ethnic groups, and then did really what by today's standards is pretty coarse genetic mapping, a couple of million markers for common variants to enable GWAS. So the first GWAS studies that appeared in 2007 were the ones that were based on the HapMap and the reagents that were built from the HapMap project. And then it was really not until like late 2000s that we started doing personal genomes. And one of the very first ones was Jim Watson. And I was on the board of 454 on the advisory board. That's one of the first next generation sequencing companies, not around now. How many of you know about 454? Pyro sequencing? Yeah, yeah. It's used a lot now.
It's 454 Roche as well.
Yeah, Roche. It's 400 gene cancer assays run on that by Thermo Fisher now. So it's not a great method, but actually it was beautifully commercialized into clinical labs. Anyway, the person who started that company was Jonathan Rothberg, and he wanted to demonstrate that you could do a whole person, a diploid genome. So we sat around after dinner and talked about who that should be, who'd be the lucky person, and chose Watson because he was sort of that time uncancelled. And so he became the first next generation genome, and we had a big ceremony here in Houston and gave it a big hard disk to him, and here's your genome. And about the same time, Craig had his genome done, but he used a lot of the data from the Celera project because he'd snuck his DNA into the program, right? And it was all Sanger-based, Sanger sequencing based. So those were the first couple of personal genomes. And then, so we started applying those to the clinic, right? And sequencing individual people. Have you seen the picture of these two kids on the van, the Beery twins? There was two children that had a movement disorder, and sequencing them revealed a very easy solution therapeutically to their malady. That was that one. And then our colleague Jim Lupsky, who has Charcot-Marie tooth disease, he's found the cause of most kinds of familial CMT, but until we sequenced him, he didn't know the one in his family, and we found that straight away. So it was these nice demonstrations of personalized genomics. And about the same time, we were working with array hybridization and these new techniques that could make inexpensive, very dense arrays. And we're using them for different hybridization experiments, but we thought, let's look for new genes. Let's take RNA and look for new genes. If we tile the genome, put RNA, we might find signals that suggest we're genes that we're not recognizing otherwise. And then we flipped that on its head and thought, why don't we try and capture DNA and do oligonucleotide capture for diagnostics. So that was the beginning of oligo capture for sequencing, and now it's a pretty standard method. So it was with that method towards around 2010 that we started to do very large numbers of people. So that's kind of almost enough history for you, right? So, oops, we're going the wrong way. So this is where we were like before the genome. I talked a lot about the, the sociology and technology, but the genetics was very naive. So we knew about a few single gene conditions, and we knew that adult genetics was more complicated, right? So it's one thing to look at cystic fibrosis. It's quite another thing to look at early onset MI, right? Someone has a heart attack, or my uncle's died from a heart attack. Maybe it's in the family, that kind of thing, right? But as you know, with those adult conditions, there's statistical evidence that there's genetic signals there, right? Like you look at twin studies and you look at primary relatives, and you can say there's a heritability to this trait, right? And so if there's a heritability, there ought to be molecules that drive it, right? I mean, and it's, you can say it might be the common households and things like that, but all of that's studied out, and indeed there should be molecules to explain the genetic risk. So when we went into doing the genome, it was really with the thought that if we could figure out sort of what was going on in genes, we could probably pretty quickly solve those genetic problems. And this is my representation of it. Like the Genome Project's like a cauldron, you boil it, and all of that stuff just falls out, right? The sequence of life, drug interactions, all that, just without a problem, right? So of course, not so simple, right? And here we are today in genetics. Like on the one hand, we've got simple Mendelian diseases, right? And how many of you are working on a Mendelian disease? It's a good thing to know. Anybody? Okay, so you know what I mean by Mendelian disease, though, right? It's like there's 20,000 genes. There's probably 10, 11,000 of them that, if mutated, cause a phenotype that's a disease in a child. And right now we know about half of them. And these methods, particularly the exome sequencing, have really driven the discovery very fast. So genes are popping out all the time. And that's the growth in Mendelian disease, right? And if you want, looking at OMIM, are you familiar with OMIM, the online? Yeah, look at the statistics page. It's pretty interesting, right? Meanwhile, over in adult genetics, after the first GWAS that was done in like 2007, like 20 years later nearly, right? It's useless. I mean, now let me be really clear. I'm saying extreme things. Of course, an enormous amount of knowledge about genetics and about function, everything has come out of genetic screens. But what we lack is really causative alleles that are in genes that you can do functional studies on and convince yourself that you've unlocked the secret of the molecules that are driving the heritability that was measured statistically. That's what's missing, right? Any of you working on in this area? Working on statistical genetics or looking at common complex disease in adults by genetic methods? Nope, that's great. Right. So have you heard of polygenic risk scores? Any of you working on polygenic risk scores? Really?
I did, yeah.
You did? You
Yeah.
gave it up? Good for you. What happened?
Well, at the NIH, I was doing polygon first story.
Yeah.
Before coming.
Yeah. Can you explain them to everybody?
I think I have a good intro explanation down about how we typically think of variants as like one variant with a large effect, but PRS score is looking at all the variants that have smaller effects and aggregating them together in a score so we can look at the impact of common variation on variability in phenotype. In one sentence.
Yeah. Thank you. That's a nice précis to the topic. Let me give you what you might call a more cynical
Okay.
descriptor. So you know how GWAS works, right? That is that you're essentially looking for markers that associate in a test group versus a control group. You get the signal from those markers, those genetic markers, more often. And then you look with statistical methods at the probability of that happening, and you assign a likelihood to it, and you say, aha, we've got a GWAS hit here, right? Like this part of the genome is somehow linked to that disorder, right? So that's GWAS. Now, and as I said, the difficult thing has been following up those individual hits. Even with fine mapping followed by trying to do functional studies has been a very hard thing to do and has not brought about a lot of success. Polygenic risk scores are essentially the flip side of that coin, where you take those hits and use the knowledge of them to aggregate the genetics of anybody to say, if you've got a signal in that area, then your polygenic risk score will be higher than a person who doesn't have a signal to contribute to that signal. So the failure of it is largely, I mean, you've heard there's always problems with ancestry but the real failure of it is it's a population-based derived statistic that you then try to apply to sort of underpinnings. I call it a triumph of statistics over common sense, right? So what am I trying to tell you? I'm trying to tell you that polygenic risk scores been very popular. They don't have the power to solve these clinical problems that people wish they did, even though you've probably heard people say wonderful things about them. Do you test all your polygenic risks and we'll find something you should worry about, right? So anyway, that's polygenic risk scores. But the real underlying problem here is we don't know the sort of the molecules that underlie the genetic signal that should be there. So how bad is it? If you have a heart attack at middle age, there should be like a 40% of the risk for that should be explainable by molecules, but only about 5% is explainable by known genetic signals. So we're really missing something in adult disease. So that's the state of genetics. Now, this is where I want to ask you to get you to ask questions, because otherwise I'm going to tell you about the somatic cell project that we're doing right now, which is kind of a different topic, right? But I would encourage you to tell me why you won't sequence your genome and put it on the internet. Right? Has anybody done that yet? Has anybody got their own genome sequenced? No? Lack of curiosity? Lack of, I mean, not that you can't be curious in other ways, but lack of curiosity about that.
Lack of protection.
Lack of protection.
Yeah.
Are you saying that you don't trust the government?
Mhm.
Nobody signed up for the All of Us program? Do you know about it?
Actually, I did.
You did?
So I do have my sequence.
Oh.
Yeah.
Did you get a genetic report?
Yes, I did.
Okay.
Yeah.
Was it interesting?
Somewhat interesting. Like, it will tell me that because of certain gene, I might not have any, maybe talk to my doctor when I take certain drugs, let's say. Yeah.
Yeah. So the All of Us program was launched. It started off under Obama as precision medicine program and kind of got morphed through different administrations. But its aim was to sequence 1 million participants. So you went down to the CVS and gave blood, or?
No, so I moved from Pittsburgh. So I was at UPMC and they were actively recruiting people willing to, take blood and willing to, kind of participate in the study, which I did.
Excellent. So the idea was to take generous altruistic participants and get their genomic sequence and a lot of phenotype data. And you can you have the option of sharing your full medical records, and they'll take other fluids and measure stuff in it, and build the All of Us workbench to allow you to access that data so you can do genetic mining studies and hopefully solve some of this dilemma in adult disease, right? So that's the All of Us program. That's one vehicle for you, one way for you to get your genome sequenced. So we've got one in 30 participate in it, right? You don't want your genome sequenced?
My friends told me that I'm not allowed to because I would know too much.
Oh, okay. That's a good one. That's a good one. Yeah. So we did me and Jim Lubsky at the same time, and mine is just so boring. Like I have to kind of make stuff up. I mean, I have a what's that gene? It's related to Usher's syndrome. I have like a missense mutation there that may have something, and my hearing's going. So maybe that's the thing, right? But that's a stretch, right? Whereas Lubsky, we found this familial CMT. So we got a New England Journal manuscript out of it. So it was a big difference there. Yeah, and yeah, I guess I share the lack of compulsion to go and get a genome sequence. I think the people who are most likely to go in that direction are older people with descendants, and but they say, well, I just want to be sure we don't have a carrier mutation in the family, that kind of thing. And then those afflicted by malignancy, I think that straight away that propels you into that arena. Well, maybe I'll go on for a few minutes and tell you about this other project, the one that we're pursuing that's pretty exciting. So, we've been dealing with this dilemma of what's genetics going to tell us, and really here, when I say here, I mean in the Department of Molecular Genetics and at the Genome Center, pursuing Mendelian disease with the idea that if we understand enough of the pathophysiology of the diseases that we can link to a single gene, we'll build up stories that will eventually build bridges to one another, and we'll understand a lot of biology through single gene genetics, right? And then maybe that'll spill over and explain some of the complex genetic disease. So here's how we've been trying to make that bridge. And let me be clear. That's one thing we're doing is building this Mendelian catalogue, but at the same time, we're using the lessons from that to look in larger cohorts for things that might explain the genetic signal. And so you see this table here. This is the things that might explain the rest of the missing heritability that we've been looking at. One of them is that it might be just extremely polygenic. Like it might turn out that these adult traits are extremely polygenic. And there's the thoughts about that have been propagated a lot by studies in traits like height, which do indeed turn out to have a lot of signals, GWAS signals, contributing to the trait, suggesting that there are a lot of genes, each one with a small effect. But that's just a few traits. You know about burden studies? Yeah, so this is the idea that a few genes will have partial impact, and in aggregate, you get a bad combination. Then you have the disorder. There was a hypothesis, the common disease, common variant hypothesis, that said sort of the same thing. That different genes, if you get the bad combination of alleles, they might on their own not be effective, but in combination, they give a genetic trait. And the rare variant, a rare disease hypothesis, or we call it the clan genomics hypothesis, where rare alleles might somehow have minimal impact in one gene, but if you get a bunch of them in a pathway, for example, you're going to aggregate and have the trait. That's burden studies, explains a few things, but not there. And then the next things are sort of structural variants, tandem repeats, and epigenetic signals are all kind of different things that you can imagine if you might not measure them properly with most kind of genome sequencing techniques, but if you got in there and really used the methods to find them, then maybe you'll get the genetic signal. And it turns out, yeah, they all help, but none of them really explain this gap. So our latest bug is this idea that somatic mutation may be a big driver of some of these common complex diseases. Now, can you spot the logic in this? This is the idea that there might be somatic mutations, not the kind that cause cancer, but that are causing some of the underlying as a contributor to some of these common complex disorders that are inherited. You see the logic, right? We're trying to explain a heritable function by a somatic process, right? So you can easily get there if you inherit a mutator that drives up your somatic mutation frequency, right? But this won't be that obvious. This must be much more subtle than that, because that would have been found. But it still remains possible that there are somatic signals that drive these disorders. Now, there's actually well-known diseases that are caused by somatic mutation, right? You probably could list them. Oh, this is just telling you you've got a lot of somatic mutations in you. So each mitosis, you get like 10 to 100 new changes, right? And think of how old some of the cell lineages are in you. Some of your cells have thousands of mutations accumulated, right? So there's a lot of opportunity for mutational variation in your somatic cells. And of course, some of them do cause cancer, but that's not what we're talking about today. So these are some of the diseases that are kind of pretty well known as somatic diseases. Have you heard of Bexus? And then Proteus syndrome, of course, yeah, right? You know, the elephant man. Yeah, so there are some of these diseases, and there's over there, these ones over here, you don't get germline mutations, they're lethal, but if you have a somatic disorder, a somatic mutation, it can cause a disease. You've seen these?
I've heard about those before. I haven't personally seen that, but I definitely have colleagues who have like some of the patients with these before.
So, and then there are other ones which there are inherited forms, but the somatic mutations can either mimic or exacerbate the phenotypes, right? So certainly there's a history of somatic mutation being involved in heritable disease, but the real extent of it's not known. So we've got this program going now to find those things, develop methods to efficiently find somatic mutations. It's actually a pretty tough problem. If you can imagine, so this is, see that thing on the right there? those of you, certainly those of you working in cancer, you're more than familiar with mutational signatures, right? So you can get a mutation signature from, like the prototypic experiment is you take exposed skin and non-exposed skin, and you do random sequence reads over each. And pretty quickly you find mutations, right? And then you look at the class of change, what bases change to what, and then you get essentially a signature, and there's catalogs of these signatures, and you can see the signature of UV damage, right, in the skin. So that's easy to get because you don't have to get the same part of the genome in the different samples. You've just got to survey each one, right? And then, so that's the top of the curve there, right? Where there are these kind of frequent events, but very low allelic frequency because these are late recent mutations that are just, you might be, just one cell has one mutation. You might pick that up in your sequence assay. The other end is the embryonic ones where, like, just immediately in a post-zygotic state, you have an early mutation, so half your cells have the mutation. So that's going to be easy to find because it's high frequency, right? But they're probably the ones we're really interested in between. They're going to be a situation where an organ has a few cells, or a cell essentially, that gets a mutation, proliferates, not cancer, but it has a change in state that can cause local disease and eventually contributes to what we are now calling an adult common disease. Right. So I've got a whole program now that is looking for that. It's called SMAT, and what this is the idea is it's a reference project that aims to get access to appropriate tissues and then develop methods to sequence and to analyze the data using the all the knowledge of developmental biology. And they've got a website, you can go and look at it, and we have a marker paper out now that you might enjoy reading that really outlines all of the different methods we're using to get that data. So it's one of these national networks. The first phase was benchmarking, figuring out how any of you use duplex sequencing methods. So this is a method where, by the molecular trickery, at the end of the day, you know if you find, you know which molecule your sequence started from. Basically, if you imagine, you take a sample before amplification, shear it, put tags on it, and then later amplify sequence, you match the tags back to the original molecule, you can tell the Watson and the Crick strand. And only a variant that appears in both is genuinely in the original molecule. Anything else is an error. So all of these kind of methods are explained and variations on them and how they can be used. But
is sampling being done at the organ level as well and multiple different organs?
yes, so I can't see my slides, but let me show you the organ map. There's 15 organs, right? I think the next one is a better. There you go. Yeah, so all three germ layers and blood and buccal swabs as well for accessibility. A few organs you'd expect to be there aren't there because these are taken from the transplant programs and it's hard to get them. And brain, there's several parts. So short answer, yes.
Amazing.
Yeah, hopefully it's useful. Are you studying cancer mutations or?
I'm a virologist and I'm just getting into virus evolution. What I've been finding is in the like previous era, people used to look at viruses as a really simple system to study evolution because the genome is small. There's only so many genes, and I'm just getting into this now, looking at how different structural proteins and non-structural proteins may have co-evolved and what sites and what does that mean for broader scale virus evolution. So I'm not doing sequencing, all post-sequence analysis.
What viruses are you mainly interested in?
Focusing on enteric viruses right now. I'm in the norovirus lab and starting with caliciviruses in general. There aren't any of those viruses, pretty small genome.
That's great.
Thanks.
That's great. Yeah, so we've just now, we finished benchmarking, getting all these methods to work. We're just now getting data from the first 25 tissue donors. And that's what I was on this conference call today, and starting to see some of the shared mutations between different tissues and the differences in individuals with background levels of mutation and effects like this. So the ultimate idea here is to build a catalogue. So when you go and study the disease you're interested in, if there's a somatic component, you'll have a reference set to go to it. And of course, we're also doing some of those disease studies in endothelial cells involved in vascularization, in endometriosis, and in the thyroid. But there's lots of opportunities there to look at somatic mutation. So I don't want to give any more formal talk, actually. I think this, like I said, I can go on a lot, but that's, I don't even want to show the rest of the slides. I think I'd rather hear from you guys about what you're working on to the extent we've got time, and talk about your work. What's your? I'll pick on you because you. Well, okay, you got out of it. There's someone in the back row.
Study coordinator, I help patients to get on different trials. And then right now I'm a program manager for the financial finance and it's really wonderful topic. I think it's very important to talk about history because we all, learn from what was before and then help us to move forward. And like you say, like the technology, there's like the technology for sequencing or computer power, it's so much different nowadays compared to before. I wonder how you feel you adjust when different significant change happen, either it's overall technology or there is like significant change in your research area. How do you adapt? And how do you like keep up learning or to utilize the resource this world has?
Yeah. Well, first of all, good for you for doing what you're doing professionally. I was trained in a cancer institute, Peter MacCallum Cancer Institute in Melbourne. The fanciest thing that came along was cisplatin. Can you believe? So and nucleotide analogues, it was such brutal stuff, that chemotherapy. And now look at what you're working with today. Such an advance. I mean, I realise we're far from being where we need to be. But your question, adaptability, we used to make fun of the guys who didn't understand molecular cloning, like the biochemist. That guy's old. He's never going to learn how to make a plasmid, right? And most of them didn't, right? And I thought that was weird. And now I'm looking at large language models and thinking about how we're going to apply some of these AI techniques to what we do. And I'm like, yeah, maybe they had the right idea. But so yeah, it's a challenge. And I don't know what the real answer is in the end. I can say that one of the reasons we've flourished as a centre, I've got to tell you this, I don't get a chance to do therapy too often. When we started the Genome Centre, it was seen as an anachronism because it was focusing on sequencing, which was going to go away. We finished the genome and people said, well, I guess you'll go home now. And then, we started doing these large scale genetic projects and people said, yeah, you're done. And then we did like the first, did cows and monkeys and mice and rats. People, well, thank you, finished now. And then we did personal genomes. And now people are saying, well, sequencing is routine in the clinic now. We don't need genome centres. So we've always had this pressure to do something new and different in order to even survive as an entity. And I don't think we'd do what we do if we didn't have that pressure. So I guess adaptation comes from environmental pressure. That's my answer. Anyone got a better one? Yeah, and so, I mean, like one of the bigger challenges right now is, of course, is interpreting genetic variation, right? I mean, there's so many VUSs and unknown variants, right?
Yeah, we joke that's our job security clinically because you always need like geneticists to put everything into context. But I agree. I think that's a very difficult aspect, and that's actually one of the things I was really curious about. Because clinically, we have a lot of patients in the hospital, many of them are very sick, that we clearly suspecting there are genetic causes, but obviously, even with the most advanced sequencing and most extensive sequencing technology on the current platform available, many of them don't have a clear causes, and even went through a lot of research-based studies, some from the house here, like the Gregor Consortium or the Chang-Zuckerberg Initiative. And still, even with that, like a lot of research bases, we still don't have clear answer for the family. But I'm just curious from your perspective, because obviously we're applying a lot of things that are in the pipeline right now that potentially will be clinically available one day. But what are some of the things that potentially may not be currently clinically available, but you feel like it's really the next horizon that with that can really open up a lot more things to help us interpret the data?
Yeah, so just sticking with diagnostics and prognostics for a moment, I think we'll have complete ascertainment so that you'll be able to predict every variant's function, right? So it sounds crazy, right? Well, back in 1985, it was pretty crazy to think of sequencing the human genome. Well, look where we are, right? I mean, and I could go on, like, you know, to think you'd have a computer in your pocket. So the idea that we can actually ascertain function, or at least some sort of functional surrogate for, I mean, surrogate to the true biological function for every variant is not unapproachable at all. And if we don't get there, we're getting to the 90% level. It's going to be doable. So you're going to have much better diagnostics, and you won't be burning your cycle. You won't have employment security unless you move on, right? You'll be the guy getting the environmental pressure who has to adapt, right? So that's for diagnostics and prognostics, right? So for predicting what it'll do. So that's I think part of the answer. And I think it's going to be true in cancer too. I mean, cancer is just a harder problem, right? Because you've got much more variation. And now therapeutics, right? So where's therapeutics going to go? And I'm probably the least qualified to really give an accurate opinion on that, but doesn't mean I don't have an opinion. Okay, I'm certainly interested in others of you. For example, if you're working on RNA therapies, but you know, you see the progress in a few disorders where you can knock down alleles in real time in children, right? And give cures. I think that we'll see a lot more of those innovative small molecules, you know, nucleic acids and other drugs driving, you know, conventional looking therapeutics. Frankly, think that the prospect for gene editing solutions to a vast sway of problems is more remote. But we'll get there. So does that kind of answer your question?
Yeah, I think that definitely touches upon, because I think like clinically, we kind of warn the family that even with the best technology right now, at best, we're probably flipping a coin in terms of whether we can find a cause or not. But I think that's sort of our understanding of where the field of genetics is. But I think that's sometimes very far away from the public conception of, oh, now that we have the sequence, now that we sequence my kid, we'll find an answer to whatever the kids are presenting us with.
Yeah, it's just paradigm is like first you describe something, then you understand it, then you can fix it, right? And we're so far just beginning to understand things, so it's a little way from that. Yeah, so what does this all mean for you? I mean, I'm thinking about all you guys because you're all like students and early career people, right? What do you do? And it's very easy to get overwhelmed, frankly, with your immediate challenges, right? I mean, you've got to get papers out, you've got to discover stuff and all of that. But I really think it's important that you at least spend a little time high in the sky, or, you know, doing high level conversations. I've got to say, as a postdoc, the laboratory I came to was unique at the time. There were 18 postdocs and I think one technician and one grad student, right? Which is an unusual demography for a lab, right? And at least 6 of us went down to the village to drink every night. Like we would come into work about 10 and we'd work till after dinner, and then I'd maybe go to the cafeteria for dinner, and then like at 10 at night, we'd go down and have drinks before we went home, and then come back the next day. But we'd always, you know, talk about a lot of things, but a lot of science. So that kind of communal thinking and, you know, thinking big and challenging stuff was really important for moving our thinking ahead. And so I encourage you to do that as often as you can.
So I'm like a first year grad student, so we're like in the process of rotating, thinking about projects. And I feel like Baylor has shown us a lot about like the rare disease field and like all these like big like genetic variants that have been discovered. And I'm kind of curious, like in your opinion, in terms of like kind of with the evolution you showed with like looking more at population genetics and statistical genetics, like, do you feel like there's a lot more that we can do with GWAS and like how, like, do you see an integration in any way in the future of like the common and rare together? And kind of just your thoughts on like how like we could use these tools to think about projects that could be like novel or brainstorming for like our PhD. I'm just curious about your thoughts on that intersection.
Yeah, I think like, so I kind of maybe sounded negative about GWAS and statistical genetics, but indeed, I mean, the signals are there to be understood and the mechanisms underlying them are there to be understood, and it's complicated, but it's not chaos, right? So I think if you can focus your work on a system that allows you to delve into GWAS signals in the context of a reliable phenotype, and you've got the ability to look at functional parameters that may impact that. I mean, the model experiment is that you find a gene, and then you look at the gene in a functional system, and you introduce some of the variants that are seen in some context, and you see that it actually makes a difference in the assay. That's how this stuff's going to be unfolded. But it's tough. It's tough work. I mean, working on a Mendelian gene for a PhD is pretty tough. And now you've really got the bull by the horns. But yeah, does that help?
Yeah. Thank you.
So, like on the topic of rare disease, right? I mean, whenever patients come in with rare diseases, and like obviously if there's a common variant, it helps narrow it down, right? And that can help like therapeutic companies, pharma companies, whatever, like to try to logistify making some type of like medication or some type of therapy to help treat it, right? But a lot of times in these rare diseases, you have different types of variants, right? And that's where you come into the realm of personalized medicine, which is gaining popularity, but not currently, according to most people, a very financial model. Do you think the future somehow, or do you have like a kind of hypothesis or a way that you think that personalized medicine can be more of an actual logistical option and not charging a patient like millions of dollars for one type of therapy, you know?
So a couple of things. One, you've heard of Regeneron, and their philosophical model is that working on rare disease is sensible because you can understand the system, and if you build therapeutics there, they will be portable over to common disease. Right? So it's kind of a different philosophy to the Mercks and the SmithKline and Beechams of the world, right? And they've been quite successful. They've been very successful. Exactly how much their success is geared to that actual philosophy versus the other smart things they've done, I guess you could talk about. But they certainly are pursuing that philosophy. So that's the idea that if you solve one of those rare diseases, you might find a situation where the therapeutics is portable. So the numbers are there to support the drug industry. I think that's one part of your question. The other part is with all these different variants that can cause, a single disease, even a Mendelian one, how do you deal with it? Well, it's going to be a case by case, but in some cases, different variants will still cause the same sort of next level of perturbation that a single therapeutic will fix. That's one answer, right? So, if you disrupt a pathway with a mutation, the supplement that cures you might not care where the stop codon was in the gene, right? So that's one scenario. But in others, you will need to know much more about the individual variant. But I think just like one day we'll know all the VUSs, one day we'll know a lot about what these allelic series do. And so we'll be able to give tailored solutions to each one.
And I think like, I mean, when people say like that, hopefully, I mean, already you've seen that like the whole genome sequencing price has defied Moore's law and everything. It's gone decently low compared to what it was, right? And I think, I don't know, I don't think it's like as much in the hospital right now for like newborn screening, like to have like genome sequences, but sometimes.
Yeah, I mean, like clinically, I think that's the next wave of where everything is, because I think all of us who are here probably had newborn screening, looking at our blood spots, and then looking at inherited genetic condition that can be detected biochemically. But there are several big consortium studies right now really geared into, let's just sequence the whole genome on the babies, and then obviously focusing on those conditions that are immediately impactful during like the childhood, during neonatal period. I mean, obviously those studies have their own caveats that we can talk about in other sessions. I think that's where people are moving towards. Like you were saying, sequencing itself, the cost has come down significantly to the point that it's not a barrier to get the sequence, but really what you do with the sequence is really the next step.
And I feel like you're kind of, I mean, it's good in a way that you're getting more data and like you're seeing all these new variants, but also it opens up a can of worms for all the new VUSs that you're about to discover. If you try to, I mean, yeah, if you do end up implementing like this and like trying to get a lot more genome sequence, you get so many more VUSs, and then the question is really how do you make those VUS, or how do you determine like the actual either pathogenicity or benignness of these VUSs?
There always seems to be a gap between when you're sitting in the lab and you're thinking about the tests you can do, and you actually do them, and the logic that you think this must be really useful clinically versus what a clinician will adopt and use. There's always like a huge gap, right? And so to me, it's important that you kind of that you know that, but that you don't get held back by that gap. So, like you'd think with exome sequencing, where you're taking these children who are on diagnostic odysseys, right, getting tortured for years without knowing what their disorder is, dragged from doctor to doctor, and you can do $1,000 test and nail it in like 40% of cases. You'd think that would be taken up like that, right? Took 10 years. So it's really, it's the established ways that medicine is practiced really prohibit very rapid transfer of technologies in general. I mean, there are exceptions. And it's not anybody's fault. It's not like a failure of the system. It's the nature of the system.
As a clinical.
Well, I think it's going to come and go right now. So what's the situation right now? There's excitement because a few disorders get solved by RNA sequencing that weren't solved by the genome. And there's some clever cellular biology to make the cells that will express that particular RNA that you can then detect the splicing defect in or the missing exon, right? So you solve a few more genetic diseases. So the knee-jerk reaction is then to have an RNA lab that will do that for these cases, right? So, and that maybe is sensible. But the futuristic view is if we have full predictability from genome sequence, we won't need to do that. And so I think I would hope that the latter proceeds fast enough that we don't have to spend a lot of time building these second.
Yeah, I agree. I think that's really an intermediate for us to understand the impact for genetic changes. I know clinically now they are, like you were saying, for selective genetic variants that we see, the lab will actually do targeted RNA sequencing experiments to try to understand is there an impact on the splicing. But then I'm sure, as many of you all are aware, because of the expression patterns is so different across the tissues, sometimes what you are interested in may not be well expressed in any of the sample type that are easily accessible on the patient. So I feel like that's obviously one barrier in terms of really applying it and solving the conditions. But then also at the same time, I know the holy grail is just taking it on the unbiased approach. Let's just do a whole RNA sequencing and see what's going on and potentially give us more information about diagnostics. But I feel like that's even further away from clinical use comparing to sort of these targeted approaches, trying to understand the impact of the genetic variants.
I was going to tell you one very quick bit of history that I think is a lesson. Particularly around the early part of the Genome Project, like when it wasn't finished, there was a huge industry of people trying to build gene prediction programs. So this would be where you take a DNA sequence and you run your program and you say whether there's a gene there or not, right? And you can do it computationally. And there were lots of different ways, and people would argue about my way is better than yours. Anyway, sequenced a whole bunch of RNA and found all the genes, right? So suddenly they were out of business. Right? And do any of you talk or work with the gene prediction program? Probably not now, right? You just look it up, right? There's a lesson there.
So I think one thing I was very curious about, as I think you sort of alluded to during your talk, is that as you transition from postdoc into having your own group and then leading into this international consortium trying to put together these human genome maps, and then obviously moving on to see other applications for that. I feel like a lot of these things require skills that we were never taught about or exposed to during our training, either as a grad student, as a postdoc, or as a clinical fellow. So I feel like, can you sort of comment on your experience? Like, how do you sort of acquire those things along the way, and then how are those things you acquire early on sort of help you facilitate sort of in the later stage of your career as well?
Yeah, I think that's a tough question for each of us, right? Like how do we get where we are and what motivated us? Where did your scientific curiosity come from? Is it genetic or is it something that happened? I remember when I was like 4 years old, some neighbor had the stereoscope glasses. He got in a cereal packet, right? The plastic things, and you got a card and you could see a three-dimensional bug, right? It blew me away. So I don't know if that was a trigger or that was just something I globbed onto because I'm some as yet undiscovered genetic combination that scientists have really matters. So that's it's a tough one. I grew up in a very modest farming environment where we didn't have much resources. So I was always able to, you know who MacGyver is? That was our daily life, right? Putting stuff together out of sticks and tape and wire and things. So then when I trained in Melbourne, we didn't have a lot of the fancy tools and reagents that I found when I got here. I remember one of my first lab meetings as a postdoc when I came to the US, somebody's experiment wasn't working. It was clearly his Nick translation reaction, but he was just using a kit that he bought from all the ingredients. And back in Melbourne, I'd been distilling phenol and doing DNAs and A concentration titrations and all of this stuff. So being in the, doing the basics there, I think is really helpful. But I think it's an interesting question why people gravitate to certain kinds of ways of doing science. But I believe you've all got something in common, which is that you have genuine curiosity. Now, I say, often talk to students and they'll say, well, why aren't you going to med school, and talk to them for a bit. And what I look for is there are some people who they want to come into the lab early the next morning to develop the film that they put on the night before. This is back in the days when you had radioactivity and you set the thing up and you had to come back the next day. I guess you want to come in early to see if your programs, or wake up early and check your computer jobs run, right? Because you're excited to see what the result will be. If you had that bug, that'll kind of carry you through being a scientist. And it's so I think what I had was no different for that thing. So another part of it is unemployability. But because you have this notion as a scientist, right, that you're kind of working for yourself, right? I mean, it's kind of not true. because you're very subjected to, you know, the administration and the market and the granting end agencies, but you're kind of self-dependent, really, right? You've got to think of your own ideas and execute them. And for some people, that's like you could never go and work for someone else, not with them telling you what to do all the time. You really have to be working on a cause or a thing that you want to do. And I suspect many of you have that. It means you're unemployable anywhere else. You just got to be a scientist. I hope that's optimistic and not pessimistic, right?
Yeah, no, I think that definitely touches upon, I think, what drives a lot of us into the science field. I guess like one thing, because I think you touch upon sort of the underlying driving forces for why we got into science and why we stay in science. But I feel like nowadays, for a lot of us who are thinking about staying in academia, it's not only about pushing the science forward. It's almost like you're running your own company, and which I feel like sometimes is the flip side of the coin that I don't think a lot of us see, a lot of us heard about. Like I was fortunate enough, I think, when I was doing my PhD, kind of see a little bit of that, like how my mentor was dealing with some of those issues. And but then I feel like getting from that limited experience, and then all of a sudden expected to be like, you're not only a scientist but also a businessman and having to run the whole thing. And I'm sure it's exponentially more complicated running a big sequencing center, and then with like more people involved. So I'm just curious, like from that sort of managing aspects, how do you feel like along the way some of the things that help?
Well, I think that you've got to have a driver of being able to convert your curiosity into shareable curiosity, right? So, you know, if someone says to you, I deserve a grant because I work really hard, right? You know, well, because I come to the lab every day, right? That's probably not going to stimulate the positive response you want. But if you can say, look, I've been thinking really hard about this pathway, and I believe I've got a way to understand more about it that can lead to a therapeutic, that's worth being supported, right? Anybody should support that idea, and I've got the credentials, I've been working on it. That'll get you success. So you've got to build your curiosity, you've got to substantiate it with logic, and you've got to be able to convert to tell that to other people. As far as the management stuff like that, two thoughts. One very wise CEO said to me once, this stuff ain't that hard. It's common sense and hard work, right? So, you know, if you want to run a big organization, all of you could do it. You just have to want to do it and be willing to put in the work. But there is another thing called the Peter Principle. Are you familiar with this? Who's heard of the Peter Principle? Oh, okay. It's a well-known term in some circles. It's the idea that you're promoted to your level of incompetence. Right. So what happens is if you do something really well, then the usual consequence is you get promoted, right? And if you do that well, you get promoted again. But then if you don't really do it so well, what happens? You stay there. So it's the concept you get promoted to your level of incompetence. So as a consequence of being pretty good in the lab, right, and with some of this stuff, I got to be a manager. I don't know if I'm a good manager.
My old boss used to say, good people keep getting promoted, but sometimes you want to keep them at where they're at, and they're going to be happy there too. That the best technician does not need to end up being a manager. They can be a very highly valued technician. I thought that he was very smart for saying that, and he ran the department that way. I think it's rare. I'm not sure if that's true.
I think that sounds like it. I admire already your mentor. Yeah, that sounds very sensible. You know, part of that though too, I believe, is have a willingness to surround yourself with people who are essentially smarter than you are, or better at their job than you would ever be, and be willing to like respect them for that and give them the latitude, you know, give them the room to do it. I mean, I actually see some managers who can do that and some who can't, and the ones who can do that, to me, have much stronger organizations. So, yeah.
This question promoted me to think about this. some topics are very esoteric. And as trainees, at least for me, I'm thinking about whatever I end up specializing in or really studying as a postdoc even, that there needs to be a market for it. That as somebody who might want to stay in academia, that topic should be fundable and looks like it has the potential to be fundable for a long time, that's probably what will get you a job. And this is all similar to what he said about running a company, that the product you're creating, which is a part of your science, should be able to get funding. And that part scares me. I'm not like you don't want to study something just because, oh, it's going to be the next big thing or like it'll probably get funded. You should do it because you really love to do it. And I think the struggle is, how do I know that this is going to have a market?
Yeah, I think you're very wise to say everything you said. That you need a level of fearlessness to pursue the careers that you all are pursuing. I mean, especially in the current climate, right?
Yeah.
So you can have your own views about what's topical and what's going to be saleable, but there's no guarantee of that, right? And also you drift from the thing that's most saleable because your own curiosity and what you want to do. It's even less of a guarantee that it's saleable. So there's a level of fearlessness that's required in science. Now, UMDs have it a bit easier because, you know, there is a you know, quite reasonably a job security, right? You can just go to the clinic. But if you want to do science, you have all those pressures as well, right? Because you'll be told, no, you should be keeping your clinical hours up. So there's a fearlessness. You know, I often see people who, this to me is the most unfortunate thing. They're doing okay in science in an academic role, and they'll go to industry because they get a 20% pay raise and they feel a bit more secure. And I mean, people have got to follow their own path, of course, but to me, there are often people who, if they just hung in there and stuck to their what their gut was telling them was interesting to follow, they'd flourish still in academia. But it's tough when you've got to pay the bills and worry about your future, right? Any of you start companies yet? I did. So along the way, I started a company with someone else, and then I sold it in around 2012, and it did fairly well. So, you know, well enough that I'm not as respectful to my leadership as I probably should be. And so that's certainly a pathway to follow, right? And so one of the advantages of the current change in the way research is being funded and the climate and everything is I think you'll have more opportunities to do that.
What do you think about your company and the licensing revenue going back to fund your own research? Obviously a conflict of interest there, but you could be your own research funding.
Yeah, well, I see some of my colleagues doing exactly that. I mean, you know, if your research is giving some exciting therapeutic and you start a company that supporting that kind of therapeutics, I mean, you've got to manage all the conflicts. That's quite doable. Then, yeah, you get to, like, in all kinds of ways, not just by paying for the research in your lab, but, you know, your lab might be sort of going along doing one thing and then you spin off a project and the company can take it to the next level. So lots of ways to leverage those two things. And you know, the academic world, the way it's funded and the way that startups and companies are funded are very different, quite complementary. We have you know, the environment in Houston is not as active in this area as in other parts of the country, but the opportunities and the materials to do it are here. So if you're inclined like this, I encourage you to go to any of the seminars or things that foster that. You know, like Rice University has student business competitions. Did you know that? You write a business plan, go over there and present it, and they'll give you a check to take it to the next level. Everyone getting tired? You want to go home and wrap your plants up and fix your leaky faucet or whatever?
I just wanted to like go back off of like what you're saying about possibly like starting your own company, right? I mean, you can have a business plan and all that, but I kind of know how academia works with like, you know, writing grants and getting like these funding, but I'm just curious if you know a little bit more about how these startups work. Like, I mean, everyone's like, oh, I'm gonna spin off a startup and all that stuff, but I'm just like, besides the logistical part of like, okay, here's our idea of what we want to do, how do you go off of like, I guess my main question is where do you get like funding from for that? Because you can't, at least I don't think you can get like government funding for like, can you for like buy like, you know, your own spin-off type company? I don't know too much. I was just interested if you know what you.
There are programs that are in actually being held back right now, but it will probably be reinstantiated. The Small Business Investment Research, what SBIR, and STTR, I think is the other one. These are for that. And they're the idea of a small company can hook up with an academic group, and the company gets funded. A little bit of the money can go to the academic group, and they build a portfolio from there. And up until very recently, each of the NIH institutes had to put a certain amount of their funds into that to stimulate small businesses. So it was a pretty good way for a small company to get going. But what I wanted to say is, you know who Richard Branson is, right? He's the guy who started Virgin Airlines and all of those things. He's a very successful entrepreneur. His business advice is that the hardest thing in starting a business is actually just doing anything. In other words, most people just go, oh, it's too hard to do. But if they actually take that step, it suddenly becomes possible. And that was actually my experience. I was talking with a colleague for quite a while about doing things, and eventually we took an idea that was pretty rote, and we just put in our own money and found a few angel investors. It was a small amount of money, and then started the ball rolling from there. So, and then we learned the way that business works. I mean, you could take a course, learn about C Corps and LLCs and tax structures and things, but it's common sense. So, but having an idea is a help. A thing that you think, this would be really neat if fill in the blank. Gosh, I was hoping to encourage you all to be academics. Maybe we shouldn't have got off on this twist.
Okay, so is there any questions for our online participants?
Not at the chat right now.
Okay.
Let me check. Yeah, not in the chat. I can ask them if they have any questions.
There's one at the back. But
Lane question. So I felt compelled today to go get sequenced. If my clinician orders this through the HGSC, should be like protected, firewalled by HIPAA, right? Like legal agencies probably can't access that readily. Like these commercial platforms, they upload to a database, from what I understand. This would be different.
Let's see. I don't know what the consent form says if you go to like the Baylor genetics labs, which are the commercial labs. I really don't know what they do. I assume they have the ability to commercialize your data de-identified. Right? So of course you all know that you can find you in the data mass, right, if you have certain other information. That's what it is. It is all otherwise protected at the level of a HIPAA protection. In the Genome Center, we don't do friends and family anymore. I mean, we have a clinical lab, right? We do projects that we make sure when people come to me and say, can you sequence me because my father just passed away with something? And we've now had enough bad experiences of genetic stories that we didn't really want to. So we say, you know what, you should really get proper genetic care.
I heard the HGSC used to do that.
Yeah, well, we were pretty fast and loose in the early days, but now we're more stringent on that.
Yeah, I think it's definitely, we definitely do have patients coming and being like, yeah, I really want our the genome sequence, or like other teams to be like, yeah, why don't we just send the sequencing? It's like a lot of times, at least maybe it will be different 10, 20 years down the road, like you were alluding to, we can look at a variant, we can know exactly what it caused, sort of prognostic purpose. But I think nowadays, just the way we sort of analyze all the data, the way the pipeline is built, it's like if you don't have a strong enough phenotyping, there's just no way for us to fish through the vast majority of the data and kind of make sense of what exactly that's going on. So I feel like that obviously that's a barrier for personalized genome sequencing down the road. And I think that's what a lot of things has been discussed. I think earlier on in the session, when we talk about newborn screening being a sequencing base, is that even though it's exciting, I think clinically we have a lot of caveats of how that would actually look like and whether a negative newborn screening actually means, because we've got certainly a lot of stories identifying new variants that wouldn't have been reported even with a current platform if we were to do just like newborn genome sequencing nowadays.
Yeah, so you've got that, and then you've got the obvious pathogenic variant that you didn't really want to know about. But that can be a factor. So guiding these things through proper skilled genetic care is in general sensible. It's a bit like if you had the first X-ray machine on the block and your friends kept coming over and you said, oh, let me X-ray your lungs. It's really fun. And then you find it. So.
That's the question.
Oh, yeah, okay. Oh, so yeah, you get it, right?
Yes. It's a terrible idea, but it stops.
So with the smart tissues, do you collect any other information besides the genomic data? Do you collect omics, peptidomics, lipidomics? Do you do comparisons? Like I see you have blood, but do you look at the bone derived, like PDMCs and just compare them? How do you all look at that data?
Yeah, so the short answer is yes for other sort of omic data, but the metadata and the other sort of physiological data are pretty sparse. So it's a clear deficiency.
I see. So like with this new data set that y'all are making, is that what so y'all don't include the omics with it? It's just genetics. It's just the genomic information, nothing else with it?
No, there is some clinical data.
Okay.
But it's quite sparse is my point.
Oh,
and I think that the consortium is learning that it would be a lot better off if it had more of that data to explain the differences. Like I just found out, so I think on my slide it says exclusion criteria, no cancer. Yeah, look, right there it says.
yeah.
So for 3 years I've been looking at that, right? And then like a week ago, then someone says, oh yeah, donor 5, they had breast cancer, and then someone else had a uterine cancer. Like, that sounds like cancer to me, right? And so, do we have the record of their treatment? No, not really, right? And so if you're looking at low-level somatic mutation, you're kind of interested to know if someone went through chemotherapy like 20 years ago, right? And so actually, it's not quite as bad as that. Someone else pulled me aside and said, these were not treated with local surgery and radiation, no chemo for any of these 4 cases. But that's, I think the project was started very much with a nucleic acid mentality in mind, and it's starting to appreciate that some of these nuances, well, not nuances, some of the necessity of the complete data. I can see people are getting worn down here.
Yeah, so like I think that's a nice point. I really appreciate you spending time with us this evening, talking about your journey through the whole genome sequencing, talk about what you envision as sort of the next wave moving forward, and share a lot of your experiences. I know as organizing committee, we have a small token of appreciation, and
Oh, that unnecessary but much appreciated.
Can take a photo.
Oh, yeah.
This is a gift to Gibbs and his certificate.
See here, 1, 2, 3. Great, perfect. Okay, thank you so much.
Oh, is this what I think it is?
Yeah, just
Oh,
from Seattle today.
Oh, you guys are great. I really appreciate that. I was like, oh no, not another glass block to put on there. This is really great. You know, I really appreciate the chance to meet you all. I think, you know, I just so much hope the best for your careers and all of that. And like James here, worked in the Genome Center. How long have you been in the Genome Center?
A little more than 3 years.
Is today the day we talk most?
Probably.
The chance to get out and talk to me. I really appreciate it.
Thank you so much.
I think we can have a group photo together.
Yeah. Am I allowed to be in the group photo? Oh, and I will frame this.
Thank you all for joining us for this lovely evening. So let's take a group photo together and you can all enjoy that. So if anyone park here at the dynamic garage, which is the garage in this building, please let me know. We have parking validations.
Oh,
If you park at the curbside, they usually don't charge after work hours, so that should be fine.
Yeah. Bye, parking.
In this building? Yeah, we have the validation of this. We have stickers like you just fill out and go out.
Was it coming over here?
I did not realize this building, and then I'm like, okay, let me just parking save them, since I know at least hours that's because no, I mean, it's like the Mendelian. Oh, it's like this next.
Extra, maybe save here.
Everyone, please, join us at the front. So you can have a group photo.